Glycolysis is one process in the largest subset of energy-producing pathways in the human body. It is responsible for the breakdown of carbohydrates to form pyruvate, which is a feeder molecule for resultant metabolic pathways. However, in order to extract energy from glucose, the cell must first invest some energy. This energy exists in the form of nucleoside triphosphates, such as ATP. Therefore, we can divide glycolysis into two phases, preparatory and payoff. Glycolysis begins when glucose, one of the main feeder molecules of the pathway, enters the cell via one of the various types of glucose transporters depending on the tissue type. These GLUT transporters allow for specific regulation for uptake of glucose. In a skeletal muscle cell, glucose enters the cell from the bloodstream through a GLUT4 transporter. Upon entry, the glucose is phosphorylated to form glucose 6-phosphate, made possible through the hydrolysis of one ATP molecule to one ADP, and prevents glucose from diffusing back out of the cell. The reaction is exergonic when coupled with the breakdown of ATP, and is catalyzed by the enzyme hexokinase, which experiences product inhibition by glucose 6-phosphate. Step 2 involves the conversion of glucose 6-phosphate to its isomer fructose 6-phosphate. This structural change allows for a symmetrical molecule in fructose 6-phosphate to enable subsequent cleavage to produce two similar 3-carbon compounds. The reaction is freely reversible, with the change in free energy being close to zero. It is catalyzed by glucose 6-phosphate isomerase. A second phosphorylation of the molecule occurs in step 3 of glycolysis, when fructose 6-phosphate is transformed to fructose 1,6-bisphosphate. This is the first committed step in glycolysis, as the products of step 1 and 2 serve as substrates for alternative pathways. It is irreversible and highly regulated. The enzyme which catalyzes this reaction, called phosphofructokinase 1, or PFK1 for short, is subject to allosteric control. It exists in two states, relaxed or R, and tense or T. When ATP, a product of glycolysis, binds to PFK1, the enzyme adopts its T state. In this high-energy state, it has a decreased affinity for fructose 6-phosphate, and this has an inhibitory effect on the reaction. The opposite is true for the R state, which is induced when ADP, or AMP, acts as the allosteric effector. In a reaction outside the pathway, the enzyme PFK2 produces fructose 6-bisphosphate from fructose 6-phosphate. As fructose 2,6-bisphosphate is the most potent activator of PFK1 through covalent modification of the enzyme, it is important in the regulation of glycolysis. When fructose 2,6-bisphosphate is bound, it reduces the inhibitory effects of ATP on PFK1. The binding of fructose 2,6-bisphosphate also allows for hormonal control in skeletal muscle cells, as the activity of PFK2 is directly influenced by insulin and glucagon-controlled protein kinases. Step 4 involves the breakdown of one 6-carbon molecule to form two 3-carbon molecules. Fructose 1,6-bisphosphate is cleaved into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme fructose 6-phosphate aldolase. Dihydroxyacetone phosphate is then converted to the feeder molecule glyceraldehyde 3-phosphate by triose phosphate isomerase. This overall reaction is reversible and is able to proceed forward in spite of its endergonic nature due to quick removal of glyceraldehyde 3-phosphate through the preceding step in the pathway. Step 5 is unique in that it is the only redox reaction in glycolysis. Glyceraldehyde 3-phosphate dehydrogenase catalyzes the transfer of two electrons from glyceraldehyde 3-phosphate to the electron carrier NAD+. These electrons will be taken to the electron transport chain to fuel the synthesis of ATP in the final step of cellular respiration. Along with its oxidation, glyceraldehyde 3-phosphate is also phosphorylated through the input of inorganic phosphate. This conserves the energy of oxidation through the formation of unstable, high-energy phosphate compound 1,3-bisphosphoglycerate. This conservation of energy ensures a net gain of ATP through glycolysis. The sixth step in the glycolytic pathway signals the start of the payoff phase. The unstable product of step 5, catalyzed by phosphoglycerate kinase 1, collapses to form 3-phosphoglycerate. This is accompanied by a release of energy, which results in the substrate-level phosphorylation of ADP to produce ATP. Glyceraldehyde 3-phosphate dehydrogenase and phosphoglycerate kinase 1 are often reversibly associated, enabling the channeling of substrate through the two reactions in quick succession. 3-phosphoglycerate undergoes isomerism to form 2-phosphoglycerate in the step 7 of glycolysis. This reaction is freely reversible and is catalyzed by phosphoglycerate mutase 1. The next step involves the dehydration of 2-phosphoglycerate to form another high-energy phosphate compound, phosphoenol pyruvate. This reaction is also freely reversible and is catalyzed by the enzyme enolase. In the final step of glycolysis, substrate-level phosphorylation of ADP again occurs, when phosphoenolpyruvate is converted to the end product pyruvate. Pyruvate kinase, the enzyme responsible for catalyzing the reaction, is subject to allosteric regulation. In most glycolytic tissues, pyruvate kinase is activated by increased fructose 1,6-bisphosphate. The enzyme is inactivated by high levels of ATP, acetyl-CoA, fatty acids, and alanine, as these molecules indicate the energy requirements of the cell have been met and slow the rate of glycolysis. The fate of pyruvate following glycolysis is dependent on the environmental conditions of the cell. In aerobic conditions, pyruvate is fed into the citric acid cycle to enable further ATP synthesis. In anaerobic conditions, however, pyruvate must undergo reduction and be converted to lactate by lactate dehydrogenase in order to regenerate NAD+ and enable glycolysis to continue. Glycolysis is an essential starting point for the production of cellular energy and is common to both eukaryotes and prokaryotes. It is important to understand this process as a fundamental example of the way in which chemical processes in our cells result in energy production.